Hi, thank you, and thank you for the very kind invitation. And I'm going to spend the next 15 minutes or so just giving you a very quick overview of academic industry collaborations and why they're so important, and particularly in the work that we're trying to do to try and find a treatment for Huntington's disease. So I work on Huntington's disease. I've worked on it since my PhD for over 25 years ago. And it's a single monogenic disease. It's the world's commonest genetic dementia. And this is a lady when she was 38, and this is her age 51, 13 years later. It's a truly devastating disease, and it affects people in the prime of life with the mean age of onset of 40. And like many neurodegenerative diseases, we have no treatment. And it's caused by a repeat expansion in the first exon of the Huntington gene, which encodes a protein, mutant Huntington, which causes disease through a predominant toxic gain of function. And so a lot of my work over the last 20 years or so has been, how do we get to the point where we can do prevention trials in this disease? And so you can have a genetic test decades before onset, if you are at risk from and you have an affected parent, to find out if you carry the gene yourself. And about 20% of people choose to have a predictive genetic test. And I've spent a lot of time studying that group. And more recently, we published a paper showing people 25 to 35 years before predicted onset. They were all in their early 20s. The brain structure and function was entirely normal. So we were very excited about this because it tells you that you are born with a genetic mutation, but it takes till you're about 25 years old, 20 to 25 years old, for the brain to start changing. And we have a big study where we're studying people decades before onset. And so this is really what we want to do. We want to do prevention trials. And prevention trials to allow us to intervene when symptoms may never occur. And in terms of, because this session is on biomarkers, I spent a lot of time, and this was a study called TRACK-HD, which I led, which really mapped the whole natural history of Huntington's disease from decades before onset to early stage disease. And we mapped it at a very deep phenotyping level. We did molecular work, genetic work, imaging, PET, cognition, which was probably the deepest phenotype of any disease at that time, that has given us huge insights. And it's now the battery that we developed is now used in all global clinical trials. We found a new genetic modifier in 2017 from the study, which is now one of the top new drug targets. So deep phenotyping studies are really important. So I'm going to give you a real whistle-stop tour of Huntington lowering. And so there are many approaches in development for Huntington lowering, and from targeting DNA with CRISPR and zinc finger proteins, antisense oligonucleotides, which Pam talked about and which I'm going to talk about today, small molecules targeting splicing, and RNAi. And all of these are in preclinical development through academic industry collaborations. But we have a lot of challenges in neurodegeneration. And the challenges we have are what to treat, where to treat, when, how. Is it going to be safe? And that's a very pertinent question in terms of what I'm going to tell you about. Is it reversible? Because we're doing a lot of gene therapy, which is a single shot. And can the dose be titrated? And we have this big challenge. This is a mouse brain, this is a rat brain, this is a large non-human primate brain, and this is our brain. And delivery and distribution to an organ the size of the human brain is our biggest challenge in therapeutic development. And so antisense oligonucleotides, and Pam has already introduced this, antisense oligonucleotides are single strands of chemically modified DNA. And they're modified to give them stability, They're able to act a bit like small molecules, because if you just give naked DNA, it gets degraded very quickly. And so ASOs bind to the cognate mRNA through Watson and Crick base pairing, and they're degraded through the RNase H1 mechanism. And that results in less of the protein being made. And so ASOs, and I became involved in this program in 2009 in a collaboration with Ionis Pharmaceuticals with Frank Bennett, because they had developed a molecule that they were interested in taking forward for Huntington's disease. So it was a very long time ago, 2009, and it was really with Don Cleveland and Frank Bennett. And ASOs, the structure of the MOE ASOs, they don't cross the blood-brain barrier. They're actually very large structures. And so we spent a lot of time trying to develop a way of how do we get them to the adult human brain. And work in non-human primates, we developed a fast push intrathecal bolus injection where you take off 20 mils of CSF, you inject the drug in 20 mils of CSF over 3 minutes, and then you get the subjects to walk for 30 minutes to distribute it, because CSF flow is dependent on blood flow. And so there was a 10-year preclinical program. It actually was started in 2005. I became partnered with them in 2009. The ASOs were effective in HD mouse models. We did a large amount of non-human primate work and also work in large pigs with long spinal cords of 52 centimeters to really test if this fast-push intrathecal bolus would get to the brain. We did work with non-human primates with pressure jackets to raise the heart rate, to see if we could increase CSF flow, because we were injecting in the lumbar region and we had to get it to the brain. So intrathecal bolus in multiple large animals gave broad distribution activity, and we did a toxicology program for 15 months in non-human primates, which, as many of you know in industry, that's a very long GLP tox study. And we identified a very wide safety margin, and this was before, this was in the, between 2011 and 2014, so it was a really very early study. And we did the first study, started in 2015. It was a first in human, 5 dose levels versus placebo, 3-to-1 active to placebo, testing this fast push intrathecal bolus with subjects walking fast. And the primary objective was, if we give this intrathecal fast push bolus, can we actually target mutant Huntington in the brain? And the study was, we tested the drug in patients with very early disease, and who were, the mean age was in their 40s. And actually, this was the result that everyone got very excited about. It was announced in December 2017, and it showed that the fast push bolus worked with the intrathecal delivery. It got to the brain, and actually there was target engagement. So it showed that the ASO got to the brain, it got to where it should do, and it lowered the level of mutant Huntington, which was the target. And this was the first study to show that in adults with a neurodegenerative disease. And so everyone got really excited, thinking, wow, well, there's going to be cures around the corner. And but this is a salutary tale. And you can see there was dose-dependent lowering of mutant Huntington. And people got very excited. It made the top story at News at 10. So the program was taken over by Roche and Genentech. And Roche were really very excited about the phase 1 data, and the community was. And they went straight from the phase 1 to a phase 3. And chose the top dose of maximum tolerated dose of 120 milligrams. And the reason for that was because it had been shown to be safe in non-human primates at the dose equivalents, and also they wanted to maximize penetration to the deep brain structures, which is the striatum. And those of you who work with ASOs or know about these molecules, similar to conjugated siRNAs, is that their distribution is very dependent on potency and dose. So the higher the dose, more penetration to deep structures was shown very clearly in the non-human primate data. So this was a really big study. It was nearly 800 people, 18 countries, testing 120 milligrams of tominersen every 8 weeks, every 16 weeks versus placebo for 2 years in patients with symptomatic Huntington's disease. And then the trial was stopped. Now, this is a really short talk for 15 minutes, and so I can talk more about this in the discussion, but it was stopped on the recommendation of the Data Monitoring Committee at 17 months. And the reason was the active group, the 8-weekly group, were doing significantly worse than the placebo. Now, there are multiple reasons, and we know a lot more about this now. The dose was too high. There was clear evidence of ASO toxicity, there was neuroinflammation, and it was too late-stage patients because they were relatively advanced stage patients because it was a 2-year trial. So there has been a huge amount of learning, and then, in fact, this is a whole topic of a talk in itself, but I'm happy to discuss further. However, so it does, and also there was spectacular mutant huntingtin lowering because everyone thought this was going to be a surrogate for clinical efficacy in Huntington's disease. And actually there was very much marked mutant huntingtin lowering, but the patients in the 8-weekly group did worse. In the 16-weekly group, they were similar to placebo. But there was a post hoc analysis, a deep dive, and I think as Pam mentioned, looking at the data to see if there was any signal. And it suggested those who are much younger, much earlier in disease, potentially had benefit. And now it's gone back to a dose-finding phase 2 study to test in younger patients, including prodromal disease, with half the dose. And that's just about to start. Also importantly, as this is a topic on biomarkers, CSF NFL went up. And at the time, Roche Diagnostics and Roche Safety said, well, it's not a recognized safety biomarker. We're not really, this was early on in the study, we're not really sure what it means. But now it is very clear, and from a number of other studies as well, that if CSF NFL goes up, take it very seriously. And if you're doing a study and CSF NFL spikes up, I would stop it early. And actually, as a community, there's been a lot of learning in this. I think the ASO study, and this is a lot of data here, will inform the future of tominersen, all Huntingtin lowering programs, and all non-ASO, non-HD ASO programs, and the manuscript is in revision. So one of the things that we're very involved in with industry partners is how do we target the mutant protein only? And the way we can do that is with a personalized genomic medicine approach. And it's targeting SNPs that are linked to the mutant allele. And we have a program with Ionis which has developed ASOs that are targeting SNPs to the mutant allele, and those are now entering into the clinic. So I talked to you about the challenges. We have big challenges in RNA therapeutics. It's a huge, it's an important thing for the UK, for UK science, and the MRC have invested in the Nucleic Acid Therapeutics Accelerator. So this is ASOs, it's conjugated siRNAs, but it's not gene therapy. So the next generation of RNA delivery needs to be able to target the whole human brain. The dose issue in the Roche study was because they were trying to get to deep brain structures by increasing the dose. So we need better drugs. We need better chemistry, backbone chemistry, stereochemistry, and conjugation chemistry. And we need better delivery. Delivery is our biggest challenge in whole brain diseases. And this is the real challenge. How do we get to therapeutics that target the whole brain? Gene therapy can only target a very small region, and ASOs really only target the spinal cord very well, which is perfect for ALS, but really the cortex, the pharmacodynamic effect is mainly in the cortex. So the MRC have recently given £8 million to an industry academic collaboration to try and tackle this, delivery and distribution. And it's a consortium with Oxford, Cambridge, UCL, and many industry partners, where we're going to try and develop new linker chemistries, new conjugates, better ways of cellular uptake for the CNS, heart, and skeletal muscle. Huge unmet need. And this is an example of academic industry collaborations. I'm a huge believer in developing drugs, but I'm a huge believer of doing it in partnership with industry. And I do think antisense oligonucleotides are going to have a big moment. I think the Tofersen study was very reassuring, and obviously the data was fantastic. And there are many different ways that ASOs can target. It's not just reducing toxic proteins, it's also targeting splice sites. You can upregulate the levels of a good missing protein like Spinraza. Lowering the levels of a toxic protein in gain-of-function diseases is just much more challenging than gene replacement for recessive diseases. And so for brain diseases, which we work on, there are many programs in development. Pam talked beautifully about Tofersen. There are trials now for C9, ataxin-2, tau, synuclein, LRRK2, spinocerebellar ataxias type 1, 2, and 3, and PRP are all in clinical development. And I think they have the potential to be a revolution in brain diseases, but we need to learn much more about them, and we need to have better chemistry. So I do want to be able to stand here in 5 years' time, possibly, and say we're doing trials in people 25 years before onset, when they're young, completely functionally normal. And that's what we want to do. But we need the regulatory framework, because 5 years ago, if I had gone to the FDA or EMA and said, I want to do this trial in 18-year-olds who carry the HD gene, there just would be no hope. And you can't do 10-year trials. So what we did was develop a staging system that captures Huntington's disease across the whole spectrum of disease. And this staging system came because of a huge amount of natural history data. And we followed the cancer staging system, stage 0 cancer to stage 4. We all know what that means. It's revolutionized cancer trials. And so Huntington's disease is defined as the presence of the gene. And it's defined from birth with stage 0, which is the presence of the gene only. And then stage 1 is a biomarker of pathogenesis, stage 2 is a clinical sign or symptom, and stage 3 is functional change. The FDA partnered us with this, and actually Billy Dunn has been a very big proponent of this, saying that this is a way that should be done for many neurological diseases. This is going to allow us to do trials in stage 2, which is the old prodromal or pre-manifest. And if we can get a positive trial in stage 2, we have a buy-in from the regulators that we could then give it in stage 0 and 1 with just biomarker evidence in a phase 4, because we know with a lot of work in the staging system, this disease is a continuum. So I do hope that we're going to be able to treat people in stage 0 20 to 25 years before onset. So that's my hope for the future. And thank you for listening, and I'm happy to take any questions. Bit of a whistle stop of a lot of...
Fantastic, sir. So much to take on board, so many lessons, and a really positive message. It can happen. Still more work to be done. Yeah. But I wonder if there's any questions from the floor. Microphone coming.
Not so much a question as a comment, Sarah. So in the Biogen ASO trial for C9orf72 ALS, very similar findings, that the dipeptide repeat proteins in the CSF came down beautifully in a dose-dependent way. Neurofilament levels went up.
Yeah.
And at 6 months, 7 out of 8 clinical features were worse, not necessarily statistically significantly worse, but all the trends were in the placebo group were doing better than the treated group. So there's a lot to learn.
There's a lot to do. I think the current MOE ASOs, I think we need more chemistry and we need better distribution. They're very pro-inflammatory, and that's interesting. I know that Biden stopped the program, and I think I would say now, and this is an example because there's industry people here, and recently Novartis did a study in Huntington's of a splicing modulator that they had repurposed called Branaplam from SMA because it was found to lower Huntington. And I was on the steering committee, and actually the trial was halted by the IDMC early, and the only reason was because NFL was going up, and it was enough of a decision to say this is bad. So I think now NFL as a safety biomarker is really something that should be taken very seriously. If it goes down, it's great. If it goes up, I think I would consider halting a study, and I think that's and I think ASOs are very exciting molecules, but there's much more work to be done, and I think that's where the NATA consortium, I hope, will have better chemistry, better delivery, because you can't just maximize the dose of these drugs to try and get more to the deep structures of the brain, because because you end up with the perfect storm of what happened with this, and also with tominersen. It was a huge study.
I wonder if I could just touch on that briefly, because there was a jump from phase 1 to phase 3, very bold move, and it could have paid off. With hindsight, do you wish a little bit more experimental medicine, work up at the early phase, might have been the better way forward?
Yeah, I hope there's no one from Roche in the audience, but I wasn't involved in that decision. So the 120 milligram dose was chosen because it looked safe from non-human primates. And this is another thing, that the non-human primate data, and actually I co-organised a Royal Society symposium last year on oligonucleotides, where all we discussed all day was oligonucleotides. And it was a big discussion with the MHRA and the head of toxicology there at the time, David Jones, is the wild type, the non-human primate GLP tox was squeaky clean. The histopathology was beautiful, massive doses, there was no histopathology and abnormal histopathology. And so Roche rightly chose the maximum tolerated dose based on the preclinical safety data. But one of these things in neurodegeneration is they're not monkeys with Huntington's disease. And so you take a dose and then you give it to subjects with a neurodegenerative disease, with neuroinflammation, with neurodegeneration, and in the tominersen study, relatively late-stage disease, and then you have this perfect storm of inflammation and NFL going up. And all our preclinical studies are done in normal monkeys and normal animals, and that isn't the disease. And so the MHRA, and in the meeting, we were all talking about going more to human organoids and brains on a chip and how we model disease better to get the tox. So the going from the phase 1 to phase 3, in hindsight, there should have been a dose-finding study. I think because it's a huge unmet need, orphan disease designation, patients are desperate with Huntington's disease, and Roche, with good faith, thought that going straight to the phase 3 was based on the phase 1 data that would potentially be a slam dunk. And it's been a huge learning, actually, in that you never can avoid and the other thing I find, and I'm not going to go on too long, but in my discussions with industry, everyone always wants to go really fast. They always say to me, Sarah, how can we do a trial in 18 months on Huntington's disease in a pre-symptomatic group, or HDISS stage 2, which is old, because we don't want to do a really long study. And I think the problem with neurodegeneration is and in an orphan disease, is that you can't cut corners and you have to do dose finding. And in retrospect, 2 or 3 years spent doing the dose finding would have been very sensible. So it's don't rush. I think that's the motto.
Thank you. Thank you so much, Sarah.